Hello, my name is Saoirse Newhouse. My actual supervisor is Professor Smythe, and we are both in the School of Biosciences. So my project was measuring the cellular response to DNA damage or stress through the design and construction of a fluorescent protein reporter for live cell imaging in cancer cells. So first I'll explain a bit about what my project was about. The whole thing was centered around ATR. So ATR is ataxia telangiectasia Rad3 related protein. This is a protein that responds to damage or stress to DNA during cell division, and it does this by activating CHEK1. This creates a downstream effect, ultimately leading to a pause in DNA replication until the DNA damage can be fixed or repaired. So ATR actually has a very similar structure and function to another protein, ATM. However, ATR responds to single-stranded breaks in the DNA rather than double-stranded. ATM is ataxia telangiectasia mutated, and this whole project is actually a mimic of a project done by the Tony Hunter lab. So everything in this project was exactly the same as in this paper by Tony Hunter, except the aim was to create the reporter for ATR instead of for ATM. So the idea of this project was that when ATR does phosphorylate CHEK1, this would cause two fluorophores to come together, creating a detectable signal which can then be viewed under a fluorescence microscope. So there were a lot of projects and problems with the project, and this is just to name a few, but weeks 1 to 4 were essentially a lot of troubleshooting and a lot of trying to deal with these problems. First, it's worth mentioning that H2B is histone 2B protein, and so this project aimed to make the fluorescent reporter for both ATM and H2B, because H2B would show us where the nucleus was in the cell. So initially, we couldn't really get ATM or H2B plasmid to transform into bacteria. It just seemed to not be working for a while. But eventually, I could get ATM to transform into bacteria, or so I thought. However, after that, I wasn't able to purify any ATM DNA from the few successful bacterial transformations that I did have. And then when I went back to the original plasmid samples, I was struggling to extract any DNA from those either. So it was suggested that the lab could have possibly been contaminated with some different strain of ampicillin-resistant bacteria, and so that's what I had growing on my ampicillin plates, rather than the ATM plasmid bacteria, but we're unsure. In the end, we had to request the samples of the H2B and ATM plasmids from the Hunter lab again, which obviously they took a little while to arrive as they were coming from America. And so at some point I was able to make successfully designed primers for CHEK1 sequences. However, after doing PCR to take out these sequences and amplify them, it turned out that these sequences were too small for DNA extraction. In terms of the kits I had for DNA extraction, required a higher number of base pairs than the DNA sequences that I had for CHEK1. It's also possible that at some point the concentration of ampicillin on the plates might have been too low, and this would have allowed bacteria without ampicillin resistance to grow on them, which is why at some point we saw satellite colonies on some of the plates. So in the end, the short project for me was more about the lab skills I could gain rather than the actual results I gained. And my supervisors felt the same way. So they moved me onto a different project, but it was essentially the same idea, but using different protein. So I was still going to gain the same skills that I'd wanted to in the end. So I managed to refine some skills I previously had, the ones I've listed here, but I also managed to gain some new skills in the end, such as macrocentrifugation, mammalian cell culture, etc. So in addition to thanking my supervisor, Professor Smythe, I want to say a special thank you to Dr. Laura Maple, as she was a big help in the lab. She dealt with a lot of my questions and queries and assisted me with certain techniques I was unfamiliar with, so thank you to her. Here's my references. Thank you.